Okay. So thank you for nice introduction and also good morning, everyone. My name is Hee-Hong Lee, and I'm Executive Vice President of GI Cell. Before I start my presentation, I'd like to thank BioKorea to give us the opportunity to introduce the company and programs that we are currently working with the immune cell expander platform. So during my presentation, in next slide, I'm going to provide more detailed explanation about how our platform can unlock future of cell therapies through invigorating immune cells to fight against cancer and inflammatory disease. Okay, so here is our forward-looking statement. GI Cell is a privately held company, and purpose of today's presentation is to provide certain information facilitating our business activity with potential partners and investors. So I'll start my presentation by providing brief introduction about the company first. GI Cell, the company, is established in 2018 as subsidiary of GI Innovation. Since then, our mission is to be a leading global immune cell therapy company and provide innovative solutions to challenges being imposed by existing therapeutics. And by doing so, we ultimately want to improve health and lives of patients and also their families. So what are current unmet challenges that we must need to address in order to provide better life to patients? So if you look in evolution of cancer treatment, you will find early generation of therapies, including chemotherapy and targeted therapy, do not lead durable clinical response in late-stage patients, although those therapies provide some survival benefit to patients in early stage. So this prompt exploration of another therapeutics, and among them, immune checkpoint inhibitors clearly demonstrate its survival benefit and also prolonged therapeutic response to certain patients, but not all. And unfortunately, majority of patients still not get any benefit from this kind of treatment. And of interest, patients who respond much better to immune checkpoint inhibitors do have higher number of infiltrating immune cells in the tumor. So this suggests immune cell therapy itself may provide therapeutic benefit to patients, and therefore immune cell therapy has been receiving so much attention recently. But as we enter advent of immune cell therapy, it still poses several challenges to be therapeutics for all patients. One of the major challenges of those immune cell therapy is actually from manufacturing process. So here we provide some of the examples of manufacturing limitation, which include use of gene editing to boost cell potency, or use of cancerous cell as a feeder cell, which may relate to compromise the functionality of those immune cells and thereby limiting its efficacy in treating solid cancers. Also, another common manufacturing limitation is related to limitation in expansion capacity and also high lot-to-lot variation in the product, all of which can contribute to increase in cell product. So with keeping all these challenges in mind, GI Cell is aimed to provide those solutions to address those challenges by developing more safe, highly potent, and scalable therapeutic immune cells from very simplified manufacturing process without using genetic engineering or cancer cells as feeder cells. In addition, we want to drive those therapeutic immune cells to target certain diseases in specific organs. Also, we further engineer those immune cells to deliver other therapeutic modality, including chemo drugs. And in this way, we believe we can maximize therapeutic benefit to patient. So we believe we can achieve all those goals using our proprietary immune cell expander platform, which will provide safe, effective, and process-efficient solution for most difficult to treat diseases that include cancer and chronic inflammation. So why we are so confident that we can achieve all those goals setting out in the previous? Because GI Cell has a team of experts who have extensive experience and also expertise in immunology, cancer immunotherapy, and protein engineering and cell therapy, together with the manufacturing process development for cells and also proteins. Moreover, our core management team has extensive pharma industry experience with a proven track record in drug discovery and development, and to get also business and corporate development. So taken together with all these combined capabilities and expertise, we strongly believe GI Cell can be readily positioned in the market and provide turnkey solution to address all those unmet needs for global community. So since our inception, our team has been working so hard and have completed so many high-value milestones, which is listed here. Among these, those milestones include obtaining core technology for the development of regulatory T cells and also NanoNK cells. Also, we successfully established many partnerships with global players to support our process development, and also with the research institute to support translational research for our upcoming clinical studies. So, with a growing market for cell therapies, I believe GI Cell is actually entering very sustainable market and close to hit inflection point as we anticipate filing of our first IND early next year for allogeneic NK cells. So as you can see, we've been achieving so many things very quickly, and our effort has been well recognized. Proving for that, the company has been strongly backed by both private and institutional investors, and so far we have raised over $75 million in combined Series A and B financing to support all those works and also future activities. So, what has been the key driver of our growth for GI Cell in addition to our people and also financial freedom? Answer to that was our proprietary platform named Immune Cell Expander Platform. So our immune cell expander platform comprises of 3 parts. We first generate ancillary proteins using protein engineering and expression technology. And then we screen those proteins by incubating with the immune cells and select those proteins giving best conditions for cell growth, viability, and functionality. After those initial selection, we enrich those immune cells with the best profile and expand them in our clinical scale manufacturing process. So as you can see from here, our overall process does not require any involvement of virus, DNA, or cancer cells. And in this way, we can avoid any safety issues related to these factors and provide more safe product to patient. Also, by eliminating unnecessary steps in the process and further simplifying that, we can lower down cost of our cell product, thereby offering very affordable cell therapies to patients. So, in addition to those features, the key feature that really setting apart our platform from others is that our differentiated platform is also a very versatile platform. So our platform can be applied to various cell types to generate different class of cell therapies. These include NK cell therapy and T cell therapy for the treatment of solid cancers, as well as regulatory T cell therapy for the treatment of non-malignant conditions, including inflammatory bowel disease. So I provide more detailed explanation about our program in next slide. So GI Cell has been working really hard to expand all those programs in the pipeline, and lead asset in the pipeline is our NK cells, called TopNK Cells. Using our immune cell expander platform, we select optimal NK cells from universal donor-sourced PBMCs. After the selection, we enrich and expand immune cells in our clinical scale process. And we have strong data from multiple runs of those processes demonstrate our platform can deliver consistent NK cell product with prolonged expansion, which enables generation of high dose and low cost product. So although our cell can be very viable in the process for a long time, our TopNK cells retain many of key therapeutic features. And most striking feature among them is that TopNK cells are polyfunctional NK cells with increased expression of CD16. And enhanced dose CD16 expression has been shown to relate NK cell synergy with therapeutic monoclonal antibodies, thereby leading to higher antibody-dependent cell-killing activity, so-called ADCC event. So consistent with the dose immune profile of our TopNK cells, when those NK cells are combined with therapeutic monoclonal antibodies, our NK cells exert more potent anti-tumor activity against solid cancers compared to each one alone, singularly alone, in preclinical settings. In addition, we believe we can use our NK cells for the treatment of hematological cancers, and thereby we can broaden our application a little bit more. Another NK cell program that we have in our pipeline is NanoNK cells. And NanoNK cell carry pH-sensitive nanoparticle that is loaded with chemotherapeutics. Because of tumor-specific homing and targeting property of our NK cells, NanoNK cells are recruited to the tumor, and low pH at tumor site prompt dissociation of those nanoparticles from NK cells, thereby inducing chemo-mediated cell killing. Also, at the same time, our NK cells in the tumor site can still engage with the tumor cells through immune synapse, and upon engagement, those NK cells release cytotoxic granule, thereby increasing cytotoxic activity against tumor cells. So as you can see, it's clear that our NanoNK cells can hit cancer cells with a double blow. One through chemo-mediated cell killing, and the other by NK cell-mediated cytotoxic activity. And in this way, we believe NanoNK cells can offer maximal therapeutic benefit to patients for the treatment of solid cancers. And also, we believe that our same technology can be adaptable to cytotoxic T cells to maximize therapeutic efficacy of those T cells against solid cancers as well. In addition to our NK cells, GI Cell is also developing personalized tumor-targeting autologous cytotoxic T cells called Express T cell. Our Express T cell is from patient-derived T cells that is primed with the engineered tumor antigen generated from our tumor antigen library. After dose priming and activation, Express T cell can be expanded in media containing ancillary proteins and tested for its potency against solid cancers. Our last cell therapy program, but not least, is our regulatory T cell therapies, named drone Treg cells. Our Treg cell express organ-specific homing receptor on cell surface, and in this specific case, that organ is the intestine, meaning that our drone Treg cell is attracted to the intestine, and at the site, Treg cells express high level of anti-inflammatory cytokines and neutralize harmful effect generated by inflammatory antigen-presenting cells and also effector T cells. Combined, those events can lead to reduction in harmful inflammation and thereby leading to repair of those damaged tissues and relieve some classic symptoms of inflammatory bowel disease. So taken together, all those data strongly support our drone Treg cell has a potential to be therapeutics against inflammatory bowel disease. So in order to advance our drone Treg cell in clinical development, GI Cell already established pilot scale manufacturing process. And from the process, we do have multiple data strongly support our platform can deliver high number of Treg cells to the scale sufficient for clinical development. So as I explained, and also as you can see from here, GI Cell has many exciting projects and programs. A leader in our pipeline is, as I told, our allogenic NK cells, which will enter clinical development next year. After that, we'll have many programs following for clinical studies, and by 2024, we believe GI Cell will become clinical stage companies. In order to achieve all those milestones and also for upcoming milestones, our team has been working really hard. However, we cannot do all this work alone. So since our establishment, GI Cell has been actively seeking partnership opportunity with the global players to support our process development and also translation and research. Now, we have more interest in partnering with the companies where we can leverage our combined capability to advance our program in clinical development and ultimately to address global need for global communities together. So these are all I have for today. And before I close my presentation, I'd like to thank everyone for your attention and also for your past and future support for GI Cells. I'll stop here to take any questions you may have. Thank you.